All right, so we're going to go ahead and get started today. Just as a heads up, I wanted to let everyone know that this webinar is being recorded, and the link to view the recording will be available in a couple of days. We'll get that posted to our website. But just wanted to let everyone know that this recording is occurring and will be available afterwards. I want to welcome everyone, and also we've got some people still joining us from the waiting room, but thank you all for being here today and engaged in this webinar. Both Scott Heisel, the president, and myself, vice president of the American Malting Barley Association, welcomes you to our webinar. This is not something we've done much of in the past, but through recent conversations with our board and our membership and then our technical committee, it was very clear that there was a lot of maybe not a lot of knowledge about where the biotech space is right now with barley, especially as there's a lot of rapid advancements in these areas with regards to barley breeding and barley improvement. And so we just wanted to open up this webinar opportunity so that we could have a much broader conversation about that. So just a few logistical things for the webinar. We are in meeting mode, as you know, just because we wanted this to be pretty interactive when we got to the Q&A part. And so we really do appreciate everyone staying on mute that is not currently talking. And totally up to you whether or not you have your camera on. If you do have questions throughout the presentation, feel free to drop them in the chat. However, it may be best to hold on to them until we're done with the presentations, just so that I don't lose them in the chat box. But certainly feel free to correspond back and forth there if you need to. We will probably just move from one presentation to the next and then just do a kind of a group Q&A at the end. And we really do encourage people to turn on their cameras and their mics then so that we can do that. The presentations will be wrapped up within the hour, but just know that we've reserved time at the end for Q&A, and that we'll have the speakers here for an additional half hour to do any Q&A that may come up. So with that, I want to go ahead and turn it over to our speakers. And I just wanted to do brief introductions of them before we get started. So first of all, in a bit, we'll have Dr. Fanli Chou joining us, and she's the Vice President for Scientific Affairs and Policy at the American Seed Trade Association, where she leads ASDA's initiatives on plant breeding innovation, intellectual property rights, domestic and international regulatory policies. Prior to ASTA, Fanli served at USDA for over 10 years, including as the Agricultural Biotechnology Advisor to the Office of the Secretary, and in positions with the Foreign Agricultural Service and the Animal and Plant Health Inspection Service. She's an accomplished policy and program leader with expertise in agricultural biotechnology, regulatory, trade issues, and multilateral negotiations. So we're really excited to have her with us today. But first, we're going to be hearing from Dr. Jason Walling, who is a research geneticist and director of the USDA-ARS Barley Malt Lab, located at the Cereal Crops Research Unit in Madison. Jason completed his undergraduate work at the University of Minnesota and spent some of that time working in the barley research plots of Dr. Don Rasmussen, many of you probably know. He completed his master's degree in plant genetics at Montana State University and his PhD from UW-Madison in the plant breeding plant genetics program. His research at CCRU currently focuses on the genetic mechanisms controlling seed dormancy and germination, including pre-harvest sprouting, and their effects on malt quality. And Jason has been a member of our technical committee since 2019, and we're really fortunate to have him a part of that. So today, Jason is going to kind of do some background introduction, discussion about the technologies, and provide some just kind of contextual case studies on how this is being applied elsewhere throughout the industry. And then Fanli will then kind of dive into the more regulatory side of this, the policies that are associated with that, and kind of even some of the consumer perception moving forward, how we can start framing some conversations around these topics. So with that, I'm going to turn it over to Jason.
Thank you, Ashley. Can everybody hear me? I'm coming up okay? Great. So again, I'm Jason Walling. I'm located in Madison, Wisconsin. Today it's 97 degrees outside and our AC isn't working in our building. So if I just slump over in my chair or fall out of my chair, hopefully one of you guys can just dive in and take over for me. I'd appreciate that. Again, just a little paperwork before we begin. The genesis of this talk sort of started in our technical committees, where AMBA is just trying to relook at their stance on where they are with GMOs and gene editing. And so I thought I would just provide a little bit of a background to the members to bring them up to speed on sort of the basic understanding of gene editing and with respect to CRISPR-Cas9 specifically. So the target audience for this is going to be generally non-scientists. So all of you, hotshot molecular biologists out there looking for advice on your CRISPRs, you're going to be distinctly disappointed in this talk. However, it will hopefully provide a general background to all of those who are really wanting to take just a little deeper dive into what CRISPR is and then how to differentiate it from GMOs. The talk will be focused in plants. I'm not going to talk at all about CRISPR use in any other systems. And again, it's the basic description of CRISPR technology. It's not an exhaustive list of all the CRISPR flavors and versions because there are a lot of them. I need to say I'm here as a federal scientist with the intent of describing the method and mechanism behind CRISPR gene editing with the hope of informing members of our stakeholder group. The talk does not reflect the agency's opinion or my own regarding the ethical nature or potential use of the technology. I will tell you that some of the slides, I introduce some mechanisms, they get a little busy. I'm going to do my best to just focus your attention on the parts that matter, and we won't worry about the parts that don't. Source information for the graphics are at the bottom of the page or at the bottom of the slide. And yes, please reserve any questions till the end. Our unit uses CRISPR quite a bit, but I will say I'm not the world's foremost expert in CRISPR. If you have questions that I can't answer or Dr. Fan Lee can't answer, I'd be happy to take them in an email and get back to you. So let's start. So just an outline of how this is going to go. We'll start off with some definitions, probably the most important slide for you guys to see. So if I pass out or if you guys pass out in between, then no worries. We'll put that one right at the top. We have to do a little molecular biology review for those of you who it's been a while since maybe college biology class, just enough so we can introduce how CRISPR is working and a little bit different than GMO. Then we'll get into gene editing and CRISPR-Cas9, and I'm going to finish off with just a couple examples of how they use the technology in wheat and barley. Okay, let's see my pointer? Okay, so just want to start out with a very simplified way of looking at the way that we geneticists and plant breeders look at improving plant improvement methods. Conventional plant breeding has been used for a long time and is sort of our go-to method. And if we want to improve a plant, generally what we would do is we would look at an elite variety, like a really nice malting barley that maybe has a characteristic about it that isn't quite working for our end product. In this case, let's just consider disease susceptibility. So what we would do is we would find another barley variety that probably wasn't very good in terms of quality, but it did have a disease resistance aspect to it. So we'd make the cross, and that first cross you would get 50% of the genome being from the elite variety and 50% from the donor variety. Well, we really just want the elite variety, and the only thing we want from the donor variety is just that resistance. So what we would do is back cross this original offspring back to this elite parent, and then select again for the characteristics that the donor would give to it. And we do this over and over and over again, called it back cross until we get almost all of the characteristics are conferred from the elite variety, and only the disease resistance comes from the donor variety. This takes a long time. I would say 8 to 10 years is short. Other ways of doing it is, instead of looking at donors from other plants in the same species, we can just look at any genome in nature. We can find a disease resistance gene from a tree. We can take that and slam it into the genome, and it'll just start working, hopefully. And that's what we call GMO, plant transgenic breeding. Another method that we can use is literally just trying to mutate the whole genome. We'll take a whole bunch of plants and blast them with mutagens, chemicals, or radiation, see which ones actually grow up. And then from there, seeing if any of them, asking the question, do any of them have any beneficial mutations that we could use to cross into the parent? Obviously, it's pretty inefficient, low precision. So not the best method for creating diversity in our germplasm. And then finally, we're looking the new editing features of CRISPR-Cas9. It's much quicker. It's considered non-GMO. And the idea is you can go right into the gene that is problematic and just fix it. So the motivation for mutational breeding, I think what most people think about is trait enhancement, making in terms of barley, whatever the traits are you're interested, how can we make them better and more able to survive in the current growing environments. And we talk a lot about resistance to biotic and abiotic stress at that point. Molly, your phone's ringing. But also as scientists, we think about something called functional genetics. And so when we're trying to understand how a gene drives a plant, we have a lot of data that sort of focuses us in on a single gene. And we think that maybe this gene would confer some sort of resistance. But the only really way to know is to just monkey with that gene, to turn it off. And so it's not so much an end product that we as scientists are interested in developing with it or using it for, but it's the idea that we can better understand some of the genes that we're looking at and how they are able to control these certain characteristics of the plants. And then another one is increasing genetic diversity. As we get to more elite crops, there is an erosion of genetic diversity because we're just bottlenecking it all. So here's some definitions. I'm sure Dr. Fanley will provide some definitions as well. So a GMO, a genetically modified organism, is the insertion and stabilization of genetic components from a non-host species. It's the insertion. This DNA comes from a non-host species, gets inserted into the genome, and it just hangs out there and it does its things. It does its thing. This is considered a GMO at that point, and there are several tests where we can look at to be able to see the gene that was inserted. Gene edited is the manipulation of a DNA sequence in the absence of stable transgenic sequences. So we go into the gene that we want to try to improve, that we think it controls a characteristic, and we edit it. And the outcome is, like this says, indistinguishable from traditional breeding techniques. Another definition I want to throw out there is there's where the source DNA comes from. Cisgenes is the insertion of DNA from a species that can normally be crossed with. You see here, we found a gene in apple and we put it in the apple. It could be accomplished with conventional breeding. In this case, a transgene is one you found in an incompatible species and we put into the species that we're interested in. So a bacteria into an apple is not conventional with, or not possible with conventional breeding. So just some examples of GMOs that you guys have all seen in the news. We have BT corn, which is a gene from this bacterium Bacillus thuringiensis, which confers some host resistance to the corn borer worm. Golden rice is from a daffodil gene, and then the Roundup Ready products and the not so flavor saver tomato all contain genes that are not from the host. Different. These are GMOs. And so a lot of people, I'm just going to take a second to talk about this. A lot of people want to know, like, how do you get these genes in the genome? Like literally, like if you're holding it, how do you just put it in? Well, so the answer is we start with an engineered piece of DNA. And it's a circle, like literally looks like this. I mean, it doesn't literally look like this, but this is how we think about it. This is a cartoon, obviously. But it's a circular piece of DNA, and it has other sequences, little pieces of machinery that allow this circular DNA to do its thing when it enters a host. One of the things we can do with this circular piece of DNA is we can put our own little sequence of interest in here. And so, again, what I want you to take away from this is this has components from different species that allow the DNA to be put into the chromosome. It gives us a method to see if the DNA got introduced into the chromosome. And so this in and of itself isn't a natural piece of DNA. And so there's two main ways of getting our little engineered piece of DNA into the host. The first one is we hijack a very natural process from agrobacterium. Agrobacterium causes crown gall. It does that by inserting its own DNA into the host cells. Like it literally sloughs off the DNA. It comes running in here and inserts into the host cell. Scientists a long time ago realized that if we attach our own little piece of DNA on here, like a gene, it'll go in there as well. The other method, which you'll see in some of the literature, is biolistic transformation. The gene gun sounds exciting, where we literally take these little pieces of DNA, put them on pieces of gold, and we just blast them right into the cell. Doesn't really make that noise, but those pieces of DNA, again, it's the same sort of process. It doesn't use the factors from agrobacterium, but at any rate, the end product is this piece of DNA gets incorporated into the cell and into the genome, and there it is. So just want to take a second to remind you that genetic material is stored on chromosomes. Chromosomes, like if you are composed of some proteins, but mostly DNA. And it's just a convenient way of packaging DNA for when DNA replicates and putting it in another cell. But keep in mind that this is the same as this. I think of it like if I was to put my foot on this corner of the chromosome and grab this one and just start pulling and unravel it, it would eventually break down or all the way unwind into DNA. But yes, again, it's just a convenient way of packaging up yards of DNA for replication. It's like having 3 or 4 yards of like extension cord. My boys would just throw it into the garage and it would be this, jumbled mess eventually. But if you wrap it up, it makes it easy to deal with. DNA is double helix. It has a top strand and a bottom strand, and the bottom strand matches the top strand. DNA can be pulled apart in this direction. These are stuck together with what are called hydrogen bonds. Not that important, but just know they're real weak. So you can just pull it apart this way, up and down, and it can stick back together. And it only sticks back together in a very specific way. These A's always have to match with T's, G's have to match with C's. I'm sure you all remember that, won't belabor that. So, and then one other thing is I want to remind you that the DNA is where it holds all of the instructions to code for our amino acids. And so if we want to create amino acids, it goes through the process of transcription and translation. And so if it needs to turn this gene on, it would start copying this gene into RNA, and then this RNA would get turned into a polypeptide. Like this is how it works. This is how it works. The order matters. The order matters. Every 3 of these guys, every 3 of them codes for a specific amino acid. ATG, well, in RNA, T's get swapped for U's, but don't worry about that. U's get swapped for T's, don't worry about that. But AUG will always, always, always code for methionine. AUC is always isoleucine. UCG is always serine. So that matters. The other thing I want to point out is in a lot of the literature, we see pictures like this. This is not a single isolated DNA. This is sort of an ellipses. It's like opening it up and peering into a window. So when you see that in some of the textbooks and literatures, that's what it means. It's, again, it's a long continuous piece of DNA. We're just looking at this little part. So what I wanted to also, because I explained that to you, because if one of these changes, one of these letters in the DNA changes, so GAA is the amino acid GLU. If that T gets swapped, or if this G gets mutated somehow and it swaps for a T, it totally changes the amino acid. And in this term, it forms a stop, which just means we're not making any more amino acid. So if one of these gets mutated, it matters. The other way that it matters is if something gets added or taken away. In that previous example, I was just showing a swap. But if something gets taken out, all of a sudden, these, like we call them triplets, these triplets get shifted. So this G is gone, AAG now is what it's reading, and it wants to make a lysine, which isn't what it's supposed to make. It's supposed to make this. And so that's the way that mutations are introduced into the genome. And it could either be, and so this one is, we call these indels, which is just short for insertion or deletion. So talking about some genome editing tools, a lot of you have probably seen these in the magazines, literature, zinc finger nucleases, talons, CRISPR, and meganucleases. What these are just enzymes that are scissors. They're really simple. All they do is cut the DNA, cut the whole DNA in half. Now remember that I told you that DNA can be pulled apart sort of north and south this way, but cutting DNA in half like this is completely detrimental to the organism. Now we'll get into that, why that's important in a second. But the other thing, the other takeaway from this is that CRISPR is, you can see this little, increasing feasibility. Talons and zinc finger nucleases, well, specific to parts of the genome, are much more difficult and more expensive to make. That's all the farther we're going to go with them. So take home message, all of these things essentially do the DNA, same thing. They find a place in the genome and they just whack, snap it in half. So what is CRISPR? CRISPR, like agrobacterium, is just something that we hijack from nature. So there was some brilliant scientists that have been following immunity in bacteria and how they're able to battle viruses coming in and attacking them. Jennifer Doudna and Emmanuel Charpentier won the Nobel Prize in 2020, and also Fang Zhang from the Broad Institute, along with a whole bunch of other scientists. I mean, it's just clearly not these three contributed to this. There's some ongoing litigation right now. You guys can, or who's responsible for it and who's a Warren patented. You guys can follow that in the news if you like. At any rate, what I just wanted to show is CRISPR is, like a lot of things in science, is an acronym. It stands for Clustered Regulatory Interspersed Short Palindromic Repeats. And the Cas is a CRISPR associated protein. So how this works is a virus would invade, this gray thing is a bacteria. And the bacteria has sort of a memory for any viruses that come and invade its cells. And it does this by taking a little bit of the DNA and it sticks it in a place in its chromosome that it holds all of these different memories of different viruses. And it does it so next time if the virus comes, it can quickly make a way, or it can rapidly degrade the virus. And the way it does that is it makes this little piece of RNA, which is very similar to DNA, and it's specific to this, meaning it's an exact copy. And so this little piece of RNA, anytime a virus, this particular virus comes into the host cell, it's going to stick to it. And it brings along a friend called a Cas protein. It's like, listen. So in this example, once the CRISPR array has already been established in the genome, if a new virus was to come, this purple one, it says, listen, I already have this memory. Cas protein, come with me. We're going to go find this thing. And the Cas protein whacks the chromosome in half or into different pieces, and it essentially destroys the viral DNA. So that's where they found it from. And they thought, if we can somehow hijack the ability to specifically break apart DNA, we might have something. Because here's the thing, chromosomes hate damage. I mean, like really hate damage. And when I say damage, I mean breaking the covalent bond, breaking the bonds east and west, not north and south, but east and west. And when it does that, organisms, they freak out. They send in the repair troops. And it happens all the time. I mean, so different ways of introducing DNA damage are things you can't see, like when it's trying to divide and replicate, ionizing and ultraviolet radiation. So, wear your sunscreen because it introduces damage to chromosomes and toxic chemicals. And when chromosomes break, again, it's detrimental to the organism. So they immediately jump into panic mode and they try to repair it. And there are two sort of ways to repair them. You don't have to remember the names of these, but the end result is when it sticks it back together, one of them is error prone. It might accidentally leave an A or a T out, or it might accidentally leave maybe 5 or 10 of the little letters out, or maybe sometime it'll accidentally add one in. The other way is a template-based repair where we can, sort of, this one's much more predictive, and we can add in little pieces of sequence that we think might be beneficial to them. So that's how it works. So I want to use a couple slides to describe the mechanism of how it works, just to give a better understanding. And so at this point, all I've told you is CRISPR-Cas9 can recognize a target part of the DNA, and then it whacks it in half. How does that edit anything? So I'm going to show the mechanism right now, but up until this point in the slide, just imagine that we've put together our vector with our CRISPR-Cas9 sequences in it, and we've inserted it into the DNA. We've shot it or used the agrobacterium, and it's just hanging out there in the cell, the CRISPR vector. And what it's doing is starting to make what are called guide RNAs. And these things are just like the little bullets that are going to go find the target sequence in the genome, and it's going to make CRISPR proteins. And so again, there are really two main components to this whole thing: the guide RNA, which is the thing that finds the target, and the Cas9 protein. And so the guide RNA is being made down here, and this is the target. It's going to make its way up here. And then what happens is the guide RNA identifies, because it's an exact match of where in the genome it wants to be, and it sticks there, and it says Cas9, have at it, and Cas whacks it in half. It cuts it up here, and it cuts it down here. That's it. That's Cas9, that's CRISPR. So how does the editing, when does the editing occur? The editing and the change in the DNA occurs during the break. Repairing occurs during, the editing occurs during the actual repair of it, which the CRISPR-Cas9 doesn't really have much to do with at all. And so just another example of it, just to walk you down. So at the top, we have our circular piece of DNA, and it could have a cornworm, anti-cornworm DNA in there. In that case, it would be a GMO, or it could have our CRISPR-Cas9. In this case, we're going to talk about it, CRISPR-Cas9, and it gets transformed into the plant cell, where it gets inserted into the gene, and then takes its action on the target. This is down here is where the actual editing occurs. But during this whole time, GMO, so transgenic plants, and CRISPR transgenic plants are all the same. They're all considered transgenic, because if you remember, that little round piece of DNA is an engineered piece of DNA. It's still hanging out here in the genome. However, once it makes the edit, you don't need it anymore. We don't need it. Because this edit should be stable in the rest of the offspring from the plant. And so we can figure out ways to get rid of this transgene or transgenic insertion. And if possible, it becomes transgene free. In plants, we can use segregation to do this. So this is basically just showing you the exact same thing I just showed you, obviously a different cartoon, but we transform the leaves with those little circular pieces of DNA that contain the CRISPR. It's going to go into the chromosome. I guess these squiggly lines in this one are chromosomes. And the transgene is going to insert itself down here, and it's going to start acting upon making the edit. In some plants, the transgene will just insert and it won't work. In some, it'll insert, but it will make the edit. So then we take them through generations. So these are still transgenic plants, even though they have CRISPR in them. We take them through generations, and we start asking the question, which one of you plants here has the transgenic tDNA, the little circular piece of DNA, and which ones have the mutated DNA? And through successive segregation, we can identify some where the transgenic DNA is segregated out, but the mutated DNA, the edited DNA, is there. We select this plant, and this plant is now transgene free. It has the gene edit already in it, and we can start using this plant. Some advantages and concern. Advantages clearly is its target site. We just mean that wherever we want, wherever we're looking at the target, it's really pretty specific at getting it. Like it's not going to make very many mistakes. They're easier to design than what I briefly talked upon, talons and zinc, and they're lower cost. They can target multiple genes at once. And I'll give an example here in a second of that. And they're non-GMO. Some concerns are off-target mutations. I said that these guide RNAs are looking for an exact match. We try really hard as geneticists to know that every single sequence and every gene in the genome, but the reality is we don't. And sometimes there may be areas where they stick and make an edit where we aren't expecting it. And we would see that in subsequent sequencing ways of seeing how it's mutated. It's limited to species level genetic material. So GMO can go to anything. If you're working in barley, I can go to a tree and try to find genetic material from that. Where in CRISPR, you're just whatever's available in the chromosomes is what you have. Gene flow. So if you're looking at heat resistance, if you want to edit a heat resistance gene in rice and it crosses with another grass, maybe you make a noxious weed that way. And then there's some data out there talking about residual presence of the Cas protein, but I've only seen a little bit of that. So 3 brief cases on when they use CRISPR. So wheat has 3 genomes in it. And so for every gene, it has 3 copies of it. And the scientists here wanted to edit all 3 genes or all 3 copies of a gene. They had some idea, did something with grain weight. And I'm showing you this picture because in almost every CRISPR manuscript, if you want to go and look at one on your own, they always show you the picture of the gene. In this case, it's 3 copies of the gene. They show you what the wild type means. Wild type, not in the sense that it's a wild plant, but this is just the plant we started with or the sequence we started with. And so this is sort of the, I guess, native sequence. Then down below, they're going to show you all of the edits that happen when they try to CRISPR on it. When you see dashes in it, that means that CRISPR, Cas9, and the repair mechanism deleted this whole chunk of DNA right here. And so if you remember way back to the triplet codons, what that would do, why this mutation would be effective in messing up the amino acids and therefore the protein. In any case, when they looked at some of the CRISPR edited products, they noticed that the grain size in some of them indeed changed. If you look at the wild type Canong here, clearly when they edited one of the copies of the gene, it increased the grain length. When they edited 2 copies of it, it increased the grain length, but also made them wrinkled for some reason. So again, it's very much a tool for learning about gene action, not just about producing products. Barley, something that's near and dear to my heart, and I'm sure most of yours, as is dormancy genes. These are 2 genes that I've been working on a lot. And so this paper was really interesting to me. Each one of these genes are on a different, on chromosome 5 of barley, but they're in a different spot. And so and we have a lot of data that says that when these genes in certain, when the sequences are different in certain species of these genes, they have higher or lower levels of dormancy. And so they were curious, like what happens if we were to edit these 2 genes? And you can see in the wild type in the Petri dish, it's sprouting like crazy. But in both of these gene edits, they're just hanging out there. They're not dead. They did further experiments to show that these are alive, but just have a heightened dormancy. So I also want to point out that when you edit a gene, it isn't just like, well, we're done. Fix the QSD1 dormancy problem. Like I'm pretty sure maltsters wouldn't like these guys as much. So it isn't just that simple. And then in wheat, there was a famous paper they came out with where they targeted the alpha-gliadin family, which is like 45 different genes, all in the wheat family. And they tried to alter all 45 of them at once. They were able to get 35 of them edited. And when they looked at the, so trying to reduce the, celiac, they noticed that there was an 85% reduction in immunoreactivity. So it was a great find and clearly was picked up by some of the news. So just a quick review about the action of CRISPR-Cas9. CRISPR-Cas9 allows editing at specific targets. The Cas9 protein just whacks the DNA in half in a way that the organism is not happy with, and so it quickly tries to repair it. That's where the mutation occurs. And then the CRISPR-Cas machinery, since it's no longer needed, can be eliminated from the host. And it's now GMO. And with that, I've gone a little long. I'm going to stop sharing my screen and turn it over to Dr. Fan Lee.
Thank you very much, Jason.
Yeah.
Yeah, Fan Lee, if you want to go ahead, and once we get the perfect.
I'll share my screen.
Yeah, thank you.
There you go.
Thank you, Jason.
So today I'm just going to take some of that science and try to put a spin on how it's viewed from a regulatory perspective. So, as you're moving from the lab to the government regulatory system, certain things gets a little bastardized. So I try to make sure that our definitions match up. So I'm here representing the American Seed Trade Association. Just a little bit about our organization. We've been around for over 100 years. We represent almost 700 members. And a lot of our members are plant breeders and seed companies, and they range in the type of products that they do research and produce. So so A to Z, so alfalfa to zucchini, including ornamentals. And they support both organic, biotech, and conventional agriculture. So our companies produce organic seeds as well as biotech seeds. And the company ranges in all different sizes, from the large multinationals that you're all familiar with to folks that actually grow seeds for home gardeners. And one of the things we focus on is actually try to create an enabling international environment for the movement of seeds. And I say movement, not just for trade, because when seed gets produced, it moves across borders quite often. So, our agenda and our priorities are member driven, I'm sure just like yours. And one of the topics that we really focus on lately is gene editing and how that is incorporated and can it be incorporated into plant breeding programs for our companies. So as Jason mentioned, from a plant breeder's perspective, plant breeders have been accumulating plant breeding tools for many, many, many thousands of years. And as we get closer to modern plant breeding, and we've gone from, say, hybrid breeding all the way to what I would view as genetically informed plant breeding. And this could be quite, not well informed with just induced mutagenesis, as Jason talked about, or it could be quite informed like marker assisted selection, where you know which certain markers are moving along with certain characteristics that you desire, and you can kind of track that from generation to generation. So in the last, I would say 30, 40 years, what Jason had talked about, we started using genetic engineering to produce as part of our breeding program, to bring in new genetic diversity into the breeding program and creating what we would call GMOs. And then the last 10 years or so, more targeted breeding using genome editing tools that Jason talked about. So this is the science of plant breeding, I would say. And then if you look at from the regulatory perspective, there's a variety of laws and regulations, both domestic and international, and also statewide, that governs how new plant varieties and their products are put onto the market. And these are technology neutral. So it doesn't matter what plant breeding methods you use. You have certain countries require variety registration. There are seed laws and seed regulations that governs how you package and sell seeds. There's phytosanitary requirements, so making sure there's no plant pests or noxious weed associated with your seed as it moves into commerce and move across borders. And there's just general environmental and food safety laws that you have to obey, right? So these are kind of the overarching regulatory oversight that over plant breeding, over seeds, and the foods that's derived from them. But in the last, I would say now 20, 30 years, on top of this overarching regulatory system, there were new laws and regulations that were directed at very specific types of plant breeding methods. So I would just highlight, there's only two. The US Coordinated framework on regulation of biotechnology, and then internationally, the Cartagena Protocol on Biosafety. And I'll talk about these in depth, right? So these two instruments came about because of GMOs. And it was viewed that genetic engineering, using genetic engineering plant breeding, created new reasons for these new laws to come into place. And I think what has happened when these laws came in, these regulations came into force, is that genetic engineering, GMOs, are only used in certain crops: cotton, soy, corn, alfalfa, and sugar beets, right? So from ASTA's members' perspective, a lot of our members are working on vegetable, they're working on fruits, they're working on horticulture like flowers. They have not been able to use genetic engineering, and genetic engineering may have been useful to introduce resistance or to introduce other new characteristics, but it has been out of their toolbox, and that is because of the regulatory hurdle, right? So I'll talk about that a little bit, and then talk about how, as gene editing came on board, the discussion that is currently occurring. So as Jason mentioned in more detail, this is kind of my really simplified thinking about what GMO is, what classic genetic engineering is. And this is taking a gene that is not within the plant's genome or within their gene pool, and putting it into a plant. So in this case, BT gene put into corn that gives its protection against plant pests, in this case, European corn borer. So this is your classic GMO, right? And the way we regulate it in the United States is that we actually didn't put out any new laws. We just said, okay, the regulatory trigger is how you make that product. So if you use genetic engineering to make this product, we're going to look at that product, how that product is intended to be used, and we're going to use existing laws to regulate it. So it's a bit of a round square peg trying to fit into a round hole in some cases. So if you create a GMO, USDA will look at it from a plant pest perspective. Is that this BT corn, is that a plant pest? Is there a plant pest risk? The question for EPA is, if it's a BT corn, is it intended to have pesticidal effects, right? So, and in BT's case, there is. So EPA look at it as a plant incorporated protectant. And they look to see how it can be used safely from a pesticide perspective, so using FIFRA. And then FDA look at foods that derive from this corn, BT corn, for example, and say, is it safe to be consumed by humans and by animals? And that's the viewpoint they look at. So USDA and EPA actually put out regulations, and FDA did not. FDA actually just put out a guidance that says, if you're going to do this, this is our general thinking about how you should do it from a safe perspective. So this is the kind of framework in the US, how we look at traditional GMOs. Internationally, there's this protocol, it's called the Cartagena Protocol on Biosafety. It's actually an international treaty under the United Nations Environmental Program. So the parent treaty, the mother treaty, is the Convention on Biological Diversity, and looking at everything that we do and how it impacts this biodiversity. So they looked at GMOs and they thought this could have a potential impact on biodiversity, and therefore they negotiated this sub-treaty, the Cartagena Protocol on Biosafety. So the objective of this is to actually ensure that there is a safe mechanism for what they call LMOs, living modified organisms, which is pretty much just GMOs, how you can safely move these across international borders and use them. So the UN, pretty much all nations of the globe is part of UN, and the protocol has 171 parties or countries that sign on to it. We, as the United States, are not party to the Cartagena Protocol on Biosafety. We're also not signatory to the mother treaty, the Convention on Biological Diversity. So for the Cartagena Protocol on Biosafety, the interesting thing is all the major growers and exporters and users of GMOs, Argentina, Australia, Canada, the United States, are not parties. The only big user, big grower of of GM corn, GM soy, it's Brazil, and that's party to the protocol is Brazil. And this has some kind of implications on how this treaty gets negotiated and implemented. But I think what's important for us as the United States is because we export most of our GM crops, is that most of our importers, our trading partners, are parties, and they have national laws that's based on this. So there are a couple terms I just kind of want you to put on your radar. One is living modified organism, and it's defined to mean any living organism that has that novel combination of genetic material that is obtained through the use of modern biotechnology. And then they define modern biotechnology, and I'm not going to read that. So just kind of keep this in mind. So most of our US export markets, so I would say our Canada and the US are the exception. They took this definition and they took these definition of living modified organisms and modern biotechnology, and they transposed it into their national legislation and national law. And most of them have a national law that defines LMOs, living modified organisms, or genetically modified organisms. And this was transposed with different degrees of fidelity. So there is some nuanced differences from country to country and what that definition looked like. So what these GMO regulations and legislation did was establish mechanisms for any time you want to use a GMO for planting, for processing, for feed or food use, there must be safety assessment approvals done before it can be put on the market. So there's food safety assessments to ensure it's safe for human consumption, feed safety assessment to ensure it's safe for animal consumption. And then if you want to plant it in that country, environmental assessment to ensure that you can plant it safely in that country. In many countries too, there is labeling requirements for food that's placed on the marketplace. So they can be voluntary, they can be mandatory, and they can be based on a threshold, say like you have 5% of GM ingredients you have to label, or it could be the type of food. It has to have actually DNA in the food for you to label it. So in the US, in the last 2 years, we implemented this bioengineered food disclosure standard where certain food that are derived from GMOs, from genetic modified crops, are going to have to be disclosed. And that just came into effect, I think January of this year was when it's fully enforced. So you may start seeing these symbols, these bioengineered symbols on some of our processed foods. So it's on this context of kind of traditional GMOs, regulatory systems around the world that people started talking about gene editing since I think 2000, right? So the difference between gene editing, the use of gene editing in plant breeding and GMOs is that most users, most plant breeders want to use gene editing to work within the plant's natural gene pool. So not crossing that sexual compatibility barrier that you're doing with, say, BT core. And just to kind of illustrate it, it's very similar to Jason's illustration. We can do this through conventional breeding, repeated back crossing of a variety with desired characteristic to your elite variety, multiple years of back crossing. With gene editing, you can streamline it and like expedite that movement of the desired characteristic if you know the gene sequence to the elite variety. And this really compresses the breeding time, right? You still have to go through many rounds of testing after you get your characteristic into your elite varieties, but it shortens that back crossing time. So if you think about this from like a variety or a plant with long generation times, like trees, this is really revolutionary, right? But in order for a lot of folks or the broader plant breeding community to use it, that regulatory hurdle is super important, both from a kind of a resource heavy reason, right? If you're a tree breeder, you cannot afford that time and expense of going through a GM regulation, but also from a market acceptance perspective. Like most people are kind of hesitant to eat like a GM tomato, for example. So there's some concerns about that as well. So with gene editing, the big question has been, is, are products of gene editing within the GMO regulatory scope, right? So with gene editing, you can do many different things, as Jason mentioned. You can do targeted deletions or insertions. You can edit the gene, so telling the repair mechanism how to repair it exactly. You can actually insert the gene. You can insert it from a gene from the plant's own gene pool, or you can insert it from outside the plant gene pool. So you can actually make GMOs. So I think countries and regulators are trying to figure out, like, where do we draw this line with our regulations? Like, where on the spectrum of things you could do with gene editing is a product GMO and subject it to regulation. And for sure, when you're inserting a foreign gene, a transgene, like if you're going to use CRISPR-Cas9 to insert BT into corn, it is clearly a GMO, and everybody's aware of that. It's these uses, the targeted deletions, targeted insertions, targeted editing, or targeted insertion of a cis gene. Are they GMOs or are they not GMOs? And that is something that all countries are kind of grappling with right now. So what asked us, we start thinking about this because from our perspective, we really want to create a regulatory environment where as many as our members, scientists, science notwithstanding, right, can use this technology, right? So if the science is there, if they can do it from a scientific perspective, then the regulatory hurdles should be low enough that as many folks can participate in this space as possible, right? So from alfalfa to zucchini, from aster, any of the flowers, they should be able to use the technology. So our position has always been that plant varieties developed using the latest breeding tools, CRISPR today, something else tomorrow, if they cannot be differentially, if they should not be differentially regulated, if they are similar or indistinguishable from varieties that could have been produced through conventional or earlier breeding methods, right? So like products should be treated the same under the law. So as I talked about, there's all these overarching regulations that governs plant breeding, governs seeds, and governs the product of seeds, regardless what kind of breeding methods you use. But this is talking about specifically the GMO regulations, right? So we should not be treated as a GMO if we don't look like a GMO. We, meaning the plants, not me in particular. So and ASTA has been working very closely with other seed organizations around the world, as well as the International Seed Federation, our parent kind of organization, to advocate this position all over the world in other jurisdictions. So the US government has actually in the last, I would say 6 years, made some progress to try to figure out how to modernize their regulatory framework with this new technology coming online. So what USDA did was towards the end of the previous administration, put out a final rule that exempted certain products if it could have been done through conventional breeding, and created some categories, and I'll talk about them a little bit, and to kind of try to really streamline and looking at what could be done using these new technologies, what could have been through conventional breeding, and where to draw that regulatory line. EPA, towards the end of the last administration, also put out a proposal that proposed certain exemptions, right? So under the current EPA rule, if you create a plant incorporated protectant through conventional breeding, for example, if you bred a virus resistant lettuce conventionally, then it does not have to go to EPA to be registered, right? So the proposed rule by EPA is saying, if you create, say, a virus resistant lettuce using gene editing, that could have been done through conventional breeding, so you're within the sexually compatible gene pool, then we also exempt it. But what EPA did was actually a little bit more intense. So, and we're still waiting to see what FDA is going to say. So just a little bit in the details here. So USDA created two categories of exemptions, right? So one is plants with traits that could have been developed through conventional breeding. So what does that mean? What could be done through conventional breeding? And I'll talk about that in the next slide. So that's the first category. The second category is plants and traits and mode of action that USDA has already reviewed. And this is actually aimed at the traditional GMOs, right? So USDA have looked at glyphosate resistant soybeans many different times, and they don't want to look at it anymore because they have not found any plant pest risk associated with that plant trade mode of action. So if you are going to create a soybean that is glyphosate resistant using, say, the ESPS gene, USDA have already reviewed it. It's no longer under, they no longer want to look at it before you put it out in the marketplace. But for the folks that want to use gene editing, the first category, this conventional breeding exemption category, is more interesting to us. USDA kind of took a very narrow approach to what they consider conventional breeding. They understand conventional breeding can do many different things, and they want to take kind of a slow approach to see which can be exempted. So they only have 3 conventional exemptions, conventional breeding exemptions. One, a change that is resultant from a cellular repair of targeted DNA break. So if you break the DNA, let it repair itself, it's exempted. Or if you create a break and change one base pair, that's exempted. Or if you already know that this genetic change occurs in the gene pool and you introduce that allele into your elite variety, so kind of a template directed repair, then that is exempted. But the problem, and for those of you working in wheat and barley, is that these exemptions are limited to diploids. So it's a change on a pair of chromosomes. So for hexaploid plants like wheat, you can't do this, right? So it's not, you can do it scientifically, but it's not exempted under USDA. But what USDA did that I thought was a little bit thoughtful and forward thinking is that they created a category that says, you can ask us if you can show us this could be done through conventional breeding, and we can add to this category. So for the future, expanding these categories of exemption of what could be done through conventional breeding. The other thing they did too is that if you think you're exempted, you can come and make sure, right? You can ask us and we will confirm whether your plan is exempted from our regulation or not. And you can actually go to their website and see where they have adjudicated or confirmed exemptions for certain products. So as I told you, EPA in their current rule already have an exemption for conventionally bred plant incorporated protectants. So if you conventionally bred a pest resistant variety, it's exempted from registration. So their proposal is to extend that sexually compatible plant PIPs through conventional breeding to breeding that occur through biotechnology. But it actually created a criteria. Your changes have to be within a certain range. You have to demonstrate expression profiles across time, across different plant tissues. And this, while on surface seems reasonable, it is completely difficult for folks that work in the specialty crop space, right? So for corn and soybeans and perhaps wheat and barley, we have very good handle on genetic information. We have good reference genomes. We have good data on expression levels for a number of characteristics for genes. But we don't have that for berries. We can't do that for strawberries or blueberries. And so this creates a burden of proof that it's very difficult and for certain plant breeders of certain plant species to meet. So that is a concern from ASTA's perspective. So this proposal hasn't been finalized, and we're hoping, EPA will review the comments and will finalize this rule in the near future. So EPA, we're still waiting to see how they are going to view this. What FDA, what FDA has said in the past is that DNA is generally regarded as safe. There's no food safety concerns. And all the things that folks have been inserting into plants like BT has been known to be safe to be consumed. So there's not really a concern, but they have created this kind of voluntary consultation, which is now, from a process perspective, almost de facto mandatory for folks to go in and consult with FDA to make sure that they are generally regarded as safe. So in 2017, FDA had come out and says, we have some questions about genome edited plants and food derived from genome edited plants. can you answer these questions for us to the general public? And people provided comments, but I don't think they have done anything since 2017. So internationally, remember international law has this trigger of LMOs, of GMOs, and there are two components to this definition. Was modern biotechnology used? And then in the final product, is there a novel combination of genetic material? So many countries around the world are taking this approach. So when you take a gene edited product, does it meet these two kind of criteria for it to occur, for us to trigger the GMO regulation? And many countries has determined that yes, modern biotechnology is used, but the end product in most cases is not a GMO because it does not contain a foreign gene and does not contain a novel combination, right? So this map is how people have been, how ASA has been tracking this policy development. And the green is countries, they're making this determination that there are certain gene edited products that are not GMO. And they have put in processes to determine when things are GMO or not GMO. And I think the big highlight is South America. All these countries are lining up how they view products as GMO or non-GMO. And if it's not GMO, it's conventional. And off it goes to the races. And I think one of the things that some general observation is that there is growing alignment internationally that certain gene edited plants are not GMOs. There's no DNA in the final product, even if it's in the kind of process. And many countries are trying to harmonize, and even traditional countries like EU and Japan that are importers of GM and very been GM hesitant has taken a much more proactive and progressive approach to gene edited products. Japan has actually notified 3 gene edited products, a gene edited tomato, a gene edited, and 2 gene edited fish. It's quite interesting. So, I think when we started this conversation, Ashley has said we're talking about the science, which is super important. You have to be able to do this from a scientific perspective. I talked about the regulation. The regulatory hurdles has to be compatible with what we're trying to do. And then 2, this whole market aspect. Are people going to be receptive to this product? And ASTA has actually done some work in trying to reach the market, to reach the general population. So we have a website called Innovature that actually talks about gene editing and plant breeding from a very kind of mass population perspective. This is not geared towards plant breeders or people in the ag space or even people in the food space. It's more geared to us with the average consumer. So we talk about gene editing, how it benefits our planet, how it can benefit our health, and how it can benefit our food supply. And so I encourage you to look at it because it tells a story about how scientists and plant breeders like Jason are using gene editing to really improve our planet, improve our health, improve our food. So I'm going to stop here and take some questions because I think it'll be good to have some discussion. I'll stop sharing.
Yeah, thank you very much. So now at this time, we would love to open it up to questions, whether for Dr. Chou or Dr. Walling. And you have options. You can either drop your question in the chat box, or if you would like to go ahead and just go off mute and ask your question, feel free to do that as well. So we will go ahead and open up now for that.
Yeah, I had a question for maybe Dr. Chou, probably. So obviously with the CRISPR-Cas9 system, you can include any cis gene and not have it be transgenic, obviously, that's by definition. But can you use that? Is there a loophole there where if you know the sequence that you're targeting, can you add a certain number of sequences, knowing that the complete sequence, can you add a partial sequence, knowing that the full sequence would then be something you wouldn't normally find in the plant? Does that make sense what I'm asking?
Yeah, I think this is where the conundrum is, right? Because what is possible through conventional breeding versus what is probable through conventional breeding versus what has been documented in the gene pool, right? And I think, from a plant breeder's perspective, and Jason, Dr. Welling can talk about this, like we're trying to find all the tools that we can to introduce genetic diversity. So I think from a plant breeder's perspective, you're trying to push that boundary of what has been characterized, right? So I think it's kind of interesting that USDA took a conventional breeding approach because you can't really define what could be done through conventional breeding, and people they're having a hard time with that. But in other countries, what they're looking at is it a novel DNA sequence? Is there a foreign DNA? And it's very much predicated on that. So if you're just changing sequences, most countries will go through their decision tree. First question, is there modern biotechnology? It goes to yes. And then second question, is there transgene? And if it's no, then it's conventional. If it's yes, then the third question is it in the final product? And if it's no, then it's conventional. If it's yes, it's GMO. So I think it's going to be, so I would say many of the cases that folks have gone through from a regulatory adjudication perspective has been very simple deletions and insertions that causes a knockout. I don't think we have actually seen any test cases for the very complicated edits just yet. But I think it's coming, and ASDA and ISF, International Seed Federation, are trying to figure out how to have that conversation with policymakers and regulators about more complicated gene edits and how it fits into their current regulatory framework, right? Because like gene insertions occur naturally, you could do that through CRISPR. How do we look at that? All right. So yeah, there's a question in the chat.
Yeah, so Kaylin has asked, do either of the presenters have a sense of how far off timeline-wise the US marketplace is from having a commercial malting barley variety for use in the brewing industry? And I think she's speaking specifically to a gene-edited variety in the US, potentially.
I don't have any feel for that. I'll defer to Dr. Fanley for that question.
Yeah, I don't have a good feel for barley in particular because I don't know what's in the research pipeline. And I think that the example Jason mentioned about, pre-harvested sprouting, you had one line that was a single mutation and one line that was a double mutation. And if you look at USDA's exemptions, only the single mutation would be exempted right now, right? You could go through their processes and ask if the double mutation is exempted. So that's a regulatory question. I don't know what the scientific question is. Like, do you have anything ready? I'm going to put in the chat. There is a EU database that just came online that looks at regulatory research in CRISPR-Cas9 plant breeding across the plant universe, and you can see what's in the research pipeline. I think for us, the ones that are closest to commercial market is lettuce, non-browning lettuce, and probably like a better tasting mustard in lettuce green. Those are pretty close, I would say, in the next year or two.
Thank you. Whatever other questions are out there?
I have a question regarding what the approval process has been in, say, places like Europe that have taken more stricter stances against GMOs in terms of approving them for not necessarily growth in the country, but use in a food product. Because if you kind of look at that map you showed from a barley sense, most of the barley exporters have taken a positive, or deregulation approach to CRISPRs, where most of the importing countries are a little stricter. So, in a future looking stance, say if those were malt barley producers and exporting to, say, countries that are a little stricter, right, how would that approval process work?
So right now, EU is going through a legislative initiative to look at gene editing in plants and cisgenesis in plants, right? So their current directive is that it's a GMO, full stop. And I think they are really looking at it from a brand new perspective, partly because of the Farm to Fork initiative and their Green Deal, right? They want to move away from conventional crop protection, but they need an alternative. So they're looking at what they call new genomic techniques and whether they can contribute to farm to fork and their Green Deal and thinking.
Yeah.
So they're actually, I would say the rhetoric, both from a political perspective and also from a general mass media perspective, has been very progressive towards gene edited products, meaning that they're going to find a way to make sure they're not GMOs. Whether there's going to be a third category of products, I'm not sure. So they're going, I think in early 2023, they will have a policy proposal for people to look at, to see what, looking at how they may view gene edited plant products. The concerning thing that I'm seeing is that aside from whether it's a GMO or not GMO, they're going to determine whether it's a good gene edited product. They want to put this sustainability kind of metrics on this. It's like, if you gene edit something that is herbicide tolerant, not sustainable, we don't like it, we may not approve it. But if you see gene edited barley that is, drought resistant, perhaps to them that's a good sustainability characteristics, and then okay, that's a quote unquote good gene edited product. So that's so EU is definitely, I would say, I'm cautiously optimistic that it's going to go in the right way. But Japan definitely is going the right way. But I don't think any exporters has gone to Japan to ask for the Japanese regulatory system to adjudicate on a imported product yet.
So to riff off that a little bit, we have another question about whether or not ASTA has actually done any research to really understand the consumer sentiment towards genetic editing. Do you have any ideas on that?
Yeah, so I think, okay, so folks who's been in the GMO space for a really long time really don't like it. They have their heckles up when people use GE to abbreviate for gene editing, because GE has always been associated with genetic engineering. So some of the old timers get really annoyed when folks do that, just so for awareness. But I think it's been really difficult to get at, right? Because it depends on how you ask the question. And I can share some of the findings that ASTA and even USDA and even FMI has done on consumer acceptance. If you ask the question, are you worried about gene edited food? The answer is yes. If you ask the question, do you want gene edited food to be labeled? The answer is yes. But if you ask them, what are you concerned about food? Gene editing doesn't come to the top of the line, right? So I think in our conversation, it's been, if you can, it's about the product. It's not about the technology, right? So if you can create a product that is really compelling, the genetic engineered or the genetic edited piece is secondary. Case in point, Impossible Burgers. They're up in front about using GMO soybeans and GMO heme to make it look bloody, and you can't keep them on the shelves, right? So it is a very compelling product. So I think a lot of the ASTA members are taking this approach. Our members are three different kinds, I would say. You have the traditional row crops that are using GMOs. They're going to use gene editing. They're producing corn, soy, cotton. They're going to do whatever they need to do. Then we have companies that are using gene editing as a platform. They are out and proud about using gene editing, and they're going to create a product that is super cool, right? Non-browning avocado, super tasty mustards, highly nutritious berries. And they're trying to market it as a product, and the technology is just enabling that product. And then we have the majority of our companies that are I would say, kind of like in the traditional produce space, kind of like barley, right? You guys have to protect your brand. You've never gone into the GM space. So everyone wants to use this technology from a plant breeding perspective, because it can really innovate their entire plant breeding platform. But they're very cautious about what the regulatory burden may look like and what the international market perspective may look like, and also what third-party verifications or third-party labeling, all of those things may look like for them. So they're looking to the kind of the gene editing seed companies to push that boundary and see how to test the market. And once that happens, then they'll figure out like whether it is it's worth the investment to get them into this space.
Great, thank you. I just have one question and then we'll go back into the chat. And Jason, hopefully you're still there. This question I think is probably more directed towards you, just with a better understanding of the industry. At the American Malting Barley Association, we are often talking about making barley a more competitive crop. That's what's really important because we're losing acreage to other crops, oftentimes that are either genetic modified or are already exploring genetic editing. And so I'm just kind of curious from your perspective, what opportunities appear to be that low-hanging fruit or opportunities for us to become more competitive as a crop through embracing some of these technologies?
Well, I think that's a great question to open up towards the group. We have a lot of barley researchers on here. So if anybody would like to chime in, I would like to hear the answer to that question.
Very diplomatic, Jason. Does anyone have anything to chime in on there?
From a barley competitiveness standpoint, you know, the most important thing and the biggest hurdle to growing barley from a producer is acceptance. So anything that can improve probability of acceptance is the biggest thing I think we need to look at. And if the barley community was interested in moving that way, that should be the first thing I would say that should be looked at. And the best touch point is going to be DON. I think both from an environmental perspective and from a producer stability perspective.
And when you're talking acceptance, just to make sure that I'm clear and that everybody else in the room understands, you're talking about accepted barley in terms of meeting the malt quality specs, not necessarily that they're accepting the technology, but that the actual quality is meeting the specs that the buyer is seeking, correct?
Barley quality, yes.
Yep. And so then acceptance, like you're talking there about specifically with DON levels.
Yes.
Does anybody, or maybe anybody aware in this group of folks actively working on that using genetic editing of any sorts?
There's been some real interesting, findings from different researchers out of, U.S. public universities about different genes that can be edited, turned up, turned down, that does confer large levels of DON resistance that are interesting targets to pursue, I believe. But the question has always been, where is the industry going, and would that be acceptable in a malt barley perspective, particularly, and to my earlier question, when we consider, malt barley is often traded internationally, and the countries that for frequent buyers are less are more adverse to some of those technologies.
Does anybody else have anything else to add on that? Kaylin actually dropped something about some potential research ongoing at Rutgers as well related to that. All right, well, I'll go back into the chat. There was another question and related back to the GE just kind of process for commercial utilization, genetic edited products used for commercial verification. So this was from Ken. Maybe I missed it because I had to step away, but what does the regulatory approval process look like for gene edited plant varieties being used commercially? Would food or beverage manufacturers have to do something different to use these plant varieties? And I know you touched on this already, but maybe just give like a high level overview of that.
So actually, I think the question, it depends, right? So I think right now it's very uncertain, especially given that malted barley and food and beverage is moving internationally, right? So in the US, depending on the variety and the edits you make, you may be exempt from regulation from a US government perspective, GM versus non-GM. But you are still going to be beholden to international, what's required in your export market. So for example, if you're going to move malted barley to Europe, or move food and beverage made from that to Europe, it will have to go through the GMO process right now because EU has not changed their directive. And if you want to plant it anywhere, it's the same thing. It matters where it's going to go and where it's going to end up. And right now, it's not easy. It's not clear. And I think that is one of the biggest hurdles across the board for downstream growers, manufacturers to uptake some of these varieties because it creates a level of difficulty that is not cost effective right now.
All right, are there other questions out there?
I just was wondering if are these slides going to be shared?
So the recording of the presentation will be shared. And so then you'll be able to see the slides there, specifically whether Fanley and Jason want to share your slides. If you're willing to do that, you can send them to me and I can send it out with the recording as well. Okay. I know you guys actually had some really nice graphics in some of those that I'm sure some people would like to be able to refer back to.
Yeah, that'd be very, very well. Yeah, excellent session. Thank you very much.
Yeah. Yeah. One other question that just came in from Tom over at North Dakota State University. His question is, how is it enforced that gene edited crops don't get mixed? And I assume when he says mixed with other conventional crops.
Yeah. See, for a lot of our companies right now, they're treating gene edited crops in their breeding program as a GMO, right? They're storing it so it is segregated from say their conventional breeding pathway. But I think it depends what happens in the regulatory space. If internationally things start to line up that certain products are not going to be GM, and you may not need to segregate. I think you always need to segregate from a breeding perspective, but like down the value chain, it may not matter anymore.
Great. Well, I will still be here for another few minutes. If there's anybody else has questions or would just even like to engage in some dialogue with our speakers today. But before a bunch more people jump off, I just want to thank everyone for actually tuning in live today. Like I said, we had nearly 100 people registered and a lot of people emailing saying, I can't make it, make sure you send me the link. So I'm excited to see kind of the broader reach that this will have. And I just want to note too, that we knew we weren't going to be able to answer all the questions today about what does this look like for the malting barley industry and specifically our end users, whether it be in brewed or distilled products. But we really just wanted to start the conversation and for you all to know that AMBA is certainly interested in this. Our membership and organization wants to be engaged in these conversations and really be thinking about what's best for the competitiveness of the malting barley crop. And so hopefully this will just start that conversation and kind of take us further as we think about how AMBA can respond to some of these kind of rapidly advancing opportunities. So I do want to thank both presenters today for coming to speak and for your time and preparing very thoughtfully for this. And with that, I'll just open it up for any other additional Q&A or discussion that folks want to have. We can leave the room open until everyone tunes off.
Thanks everybody for your time today, and I'll do the best to get my slides to Ashley. So if any of you would like to review them, use them, you're more than welcome to them. And Ashley has my email if anyone has any follow-up questions. Thanks.
Yep, I put my email in the link if you guys have follow-up question about regulations and stuff like that. The only thing is the map that I have up there, it changes constantly. So if you want to use that map for any reason, just reach out to me so I can make sure you have the most updated one.
Great, thank you very much.
Thank you. Well, thanks everyone and have a wonderful afternoon.